So you set to go? We're recording. Okay, tomorrow we will go over the lab exam, and you will have time to finish up your skin, nose, throat thing. The write-up, it's not much to do, just answering questions from those, is due Wednesday, but you should be able to finish it tomorrow if you, because it's probably only going to take about 20 minutes to go over the lab exam. Okay? Pardon me? Why did you send me an email yesterday? Because I threatened to do that to get you to read the board. So I just wrote that on the board, and if you read the board, you got 2 points.
I sent that to you, and I was like, I probably missed something, and there's explanations attached, and I was really confused. I was like, there's no way you would just have it.
So put the micro lab exam in the subject line, that's all you had to do. Okay, we're going to start a new topic, which I think will be of interest to many or most of you. We're going to be talking about infection and disease. We'll spend some time talking about the relationship that microorganisms have with the human body, and really concentrate on the parasitic relationship, which may but doesn't always result in disease. We'll talk about the offensive capabilities of microorganisms that makes them effective in causing diseases, and then we will end the semester talking about some human defense systems and applications for that. Okay? So, any idea how many cells make up your body?
1 billion.
More than that. Cells are associated, make up your body. Estimates indicate about 110 trillion. How many of those are yours?
1 billion.
About 1 trillion. So your body is about 1 trillion human cells and about 100 trillion microbial cells. We are mostly bacteria in terms of number. Okay. For a long time, people really just kind of thought, well, okay, those are just contaminants and they really don't have any real significance. In 2007, a very interesting project started called the Human Microbiome Project. This is kind of the microbiology equivalent of a similar project that was done in genetics. What was the genetics project called? Human Genome Project. The goal of the Human Genome Project was to sequence all human genes. Okay. What's the purpose of the Human Microbiome Project? To try to identify all the bacteria that are associated with the human body. And they tend to break this up into body regions, and so one group is working on the skin, one group is working on the urinary tract, one group is working on the gut, and the one that really is going, it's going to happen in stages, it's going to take a long time to do this. The one that's really going strong right now is the gut project, and trying to characterize all the microorganisms associated with your guts. Okay? And we're finding out some really interesting things. And many of those were described in that article that I said you should read the very first day of class. And if you haven't read that article, it's on the website. But it really does describe some interesting things about what is the outcome of that relationship between those 100 trillion microbial cells and your body. Because those microorganisms can influence your physiology, your state of mind, your weight. There's all kinds of things that we are now attributing to these microbial associations that really had just escaped us until we started digging into it and looking at it. One of the technologies that has really helped this project is an approach called metagenomics. And I referred to that when I talked about the diversity of microorganisms a little bit. And I said that we used to think that there weren't many microorganisms living in seawater, that ocean water was kind of a biological desert. Well, the problem is that many of the organisms that actually live there, that are microbial, we just can't culture effectively in the lab. So how do we know we're there? We look for their genetic fingerprints in samples that we take. We do PCR and we do gene sequencing. And all of a sudden we're seeing all of these unique gene sequences that are different from other gene sequences. So we infer the existence of organisms even though we can't grow them effectively. A lot of that kind of research is being done in the Human Microbiome Project as well. Well, we have talked about microbial associations already in this class. Okay? Microorganisms rarely exist by themselves in nature. They are either in mixed cultures and they coexist with other microorganisms. They may lead independent lives, but they're still mixed up with other bacteria. But sometimes there are actual relationships between microorganisms and hosts. We call those particular relationships symbiotic relationships. So microorganisms can live in non-symbiotic and symbiotic relationships. What does the word symbiosis literally mean? What does bio mean? Life or living. Sym means together. So a symbiotic relationship occurs when two organisms live together. Okay? And there are three subcategories of symbiosis, which I'm sure you've all heard of before: mutualism, commensalism, and parasitism. If we take the most simple relationship, which would be two organisms, and give it a little shorthand, where plus indicates a benefit from the relationship, 0 indicates no effect, doesn't matter if the relationship's there or not, and negative indicates harm, then mutualism would be designated as what? Plus plus, right? Both partners benefit. What context have we discussed a mutualistic symbiotic relationship with bacteria already in class?
Our stomach, GI tract.
Our GI tract, yep. How about legumes? Okay, remember nitrogen fixation? Bacteria can convert the N2 in the atmosphere to ammonium, and the plants can use that to make proteins and nucleic acids. What do the bacteria get out of the relationship? What do you get by living in a plant root? Nutrition, carbohydrate, okay, photosynthesis. Okay. So, mutualism, both parties benefit. And yes, there are some important mutualistic relationships. We've already alluded to some of them. Your gut bacteria, your good bacteria. They can keep the bad bacteria in check. What do we call that phenomenon? One inhibits the other. Antagonism, microbial antagonism. Okay? Commensalism, how's that going to be denoted? Plus 0. One benefits, the other not harmed. One of the things you did in lab is you looked at possible commensal organisms on your skin, specifically looking for staph epidermidis. What does staph epidermidis get out of the relationship? Nutrients, dead skin cells. Okay. Are you harmed by the staph epidermidis? No. Okay. Parasitism, how do we characterize that? Plus minus, right? One benefits, in this particular case, the parasite. One is harmed, and that is the host. Some of you may have potentially parasitic Staph aureus on your skin, which under the right conditions could cause damage. Okay? Well, the relationship we're really going to concentrate on is the parasitic relationship. So we want to look at that host-parasite relationship. and see what's going on. So we're going to discuss some concepts like the difference between your normal flora and your transient flora. We're going to discuss some concepts like the difference between contact, infection, and disease. One of the things that happens in parasitism is there is a competition going on, and it's typically a competition for nutrients. That competition may benefit one and not harm the other, therefore it is a commensalistic relationship. It might benefit both, but if one is harmed, then obviously we have a parasitic relationship. Does that necessarily result in harm? No. We can have situations where we actually have a disease state or an infectious state. And a person can have an infection and not really know it. Why? Because there are no overt symptoms. You may know what we call that when you actually have an infection, but there are no symptoms. Asymptomatic, or sometimes it's called subclinical infection. Okay. There's kind of just a balance that's been struck between the parasite and the host is not harmed. But if the balance tips more toward the parasite, then that infection can result in damage to the tissues, and we have a state of disease. There's a term that you'll see later on that I thought I'd throw up here right now. And that term is morbidity. That term is used a lot in epidemiology, and it refers to the incidence of disease in a population. But for right now, if you want to think of kind of morbidity as kind of a synonym for disease, that's fine. If the balance gets tipped and the parasite actually causes disease symptoms, what do we call that parasite? It's a pathogen. Okay? If the parasite kills its host, is that a good relationship? No, because what does the parasite have to do? It has to find another host. Okay? So parasites that actually kill their host are oftentimes referred to as being inept parasites. Ebola is a great example. Ebola is an inept parasite. And it kills the host so rapidly that many times there is an opportunity for a transfer of that parasite to the new host. That's why Ebola infections oftentimes just kind of burn themselves out. Okay? Is the body a good place for pathogens to grow? Potentially. Okay? Think about the human body for a second and the homeostatic mechanisms that we have. We have homeostatic mechanisms to regulate our internal body temperature. We are a perfect 37 degrees Celsius incubator, right? Okay? So any pathogen that can live within that mesophilic zone where the peak is 37 degrees centigrade, the body's a great place. But we're not just an incubator. We also provide food. Okay? And we have a circulatory system that helps to deliver those nutrients to all the locations of the body. What other factors affect growth besides temperature and nutrients? pH. Do we regulate pH in our body? You bet we do. Okay? We have, for example, bicarbonate buffering systems in our blood. Okay? So the body is really a great incubator. Is it a uniform environment? No, there are also all kinds of little microhabitats within your body. For example, is the pH of the stomach the same as it is the small intestine? No, stomach's very acidic. Small intestine tends to be slightly basic in pH. Okay? So we have all these little microenvironments, and so it's really not surprising that microorganisms grow in a number of different locations in your body, and because different species have different requirements, then there are going to be little microbial populations associated with this part of the body, a different population associated with this, etc. And that's what the Microbiome Project is kind of trying to figure out. Well, all of these relationships that involve parasites begin the same way. They begin with contact. Okay? Contact can result in a permanent colonization, where the microorganisms just take up residence and live on you or within you. If that relationship is permanent, that's what we call our normal flora. Sometimes things come and go. They're kind of visitors. They come and visit for a few days and then leave. Maybe they're removed by competition with your normal flora. Maybe they're removed by cleaning, for example. Okay? That would be your transient flora. If organisms are able to cross the lines of defense, and that's something we'll be talking about later on, then they can penetrate into the tissues and invade. And when that happens, we say we have an infection. What's one of your primary barriers against infection? The skin. Okay? And the skin is really a pretty great barrier. We'll talk about characteristics of this later on. But if you get even a minor cut or a burn and you compromise that particular barrier, it's pretty easy for microorganisms to invade and cause infection. Infection refers to the multiplication of pathogens in your tissues. Bacteria are multiplying. Well, what happens? Well, oftentimes your human defenses, your immune system, holds those pathogens in check, and maybe will do so in such a way you will never suffer any symptoms. So you'll have an asymptomatic infection. But oftentimes, those particular pathogens cause damage to the tissue. And that leads to dysfunction, a state of disease. And if that dysfunction is too severe, mortality or death can occur. Contact typically does not result in infection. That is a testament to your immune system, your body's defense system. Infection usually is resolved before it causes mortality. That is also a testament to the functioning of your immune system. Is all disease caused by infection? Give me an example of a disease that is not caused by infection. Pardon me? Cancer? Okay. We used to say heart disease, but there is some implication now that there might be infectious agents that cause inflammation, which are contributing to coronary artery disease. These are some sites of your body that normally harbor resident populations of bacteria. And if you read your book and read through the chapter, it will go through these various regions of the body and give you an idea of kind of what's living there and what you are coexisting with. It's kind of interesting to read that stuff. I'm not going to hold you responsible for all of that detail in an exam, but you know, reading that is part of getting to know yourself. What are you coexisting with? What's living with you? So there's a very robust normal flora associated with those regions. Sometimes visitors come into those regions, the transients. And sometimes those organisms, under the right circumstances, can be pathogenic. Those organisms are referred to as being opportunists. What conditions might lead to an opportunist actually causing a disease? Lowered immunity is a big one. Okay? That's really how HIV was first identified, was that HIV patients were all of a sudden showing a very high incidence of diseases that were just uncommon, things that people normally didn't get. And they started to look at it and they went, wow, I mean, healthy people have these same bacteria, but they're not getting the disease. What's going on? They have a state of lowered immunity. And the opportunists were now able to thrive and flourish and cause these kind of unusual and rare diseases. We'll talk about, later on when we talk about your immune system, why it is not uncommon for college students to get sick at the end of semesters, particularly after finals. Why? Because you're under a lot of stress. Stress produces cortisols, and cortisols depress immunity. There are a lot of other factors that can influence your state of immunity, and we'll talk about some of those later. But diet and hormones, there are all kinds of things that can affect your state of immunity. And under the right conditions, then these opportunists could get out of control. Several of you are finding that you are at least occupied transiently, if not permanently, with Staph aureus bacteria. But do you all have staph infections? No. Those particular transient flora are normally held in check, and you usually don't get sick. But under the right state of circumstances, they can cause problems. So here's just an electron micrograph of some bacteria that are part of your normal flora that are colonizing and sitting on the surface of some, I think these are epithelial mucosa cells, intestinal cells. Some interesting studies have been done that really point out the importance of your normal flora in your body's function. And these studies come from raising azenic animals. These are germ-free animals. Okay? Humans and other animals that are placental and surrounded by an embryonic sac are germ-free. Okay? As soon as you start to make that passage down the birth canal, you start acquiring microorganisms. The normal flora begins establishing right at birth. It goes through an interesting fluctuation and evolution as you age. And so the normal flora of a young child is not the same as the normal flora of a teenager or an adult. But it's something you acquire and that develops over a period of time. But what are the consequences of being delivered into a germ-free environment and developing without your microorganismal partners? There are a number of effects. And some of those are illustrated for you over here on this particular slide. You see on the left some of the effects of being raised in a germ-free state, and on the right some significance or inferences from that. So it appears that from studies of azenic animals, the microorganisms are needed for normal intestinal development. That they're significant sources of nutrition, including vitamins. Very, very much demonstrated that microorganisms are needed to stimulate proper development of the immune system. There is an interesting hypothesis. Called the hygiene hypothesis. Anybody heard of this before? This hypothesis is an attempt to explain why we're seeing incidents of certain kinds of illnesses, particularly things that are autoimmune or conditions like asthma. Okay? When I was growing up, you never saw kids at sporting events with inhalers. Now it's relatively common. The incidence of childhood asthma is definitely on the increase. Okay? There are some people who argue that the reason that we are suffering from more of these autoimmune type of conditions is that we are growing up in too clean of an environment. The children are not being challenged by an appropriate number of microorganisms, and therefore their level of immunity is not as strong as it could be. Okay? We tend to be kind of obsessed as a society by cleanliness. And we use antibacterial soaps, for example, when it might not really be needed all the time. I think it's justified in the lab here because you're exposed to so many potential pathogens. But at home, I mean, you really have to wash your hands all the time with antimicrobial soaps? Probably not. Okay? Is it really a problem if little kids eat dirt? No, it's probably good for them, at least according to the hygiene hypothesis. Okay? Taylor, we're good.
So you're at increased risk in this aspect if you have that C-section, and those are also increasing.
I don't know. I mean, that would be an interesting study to do, is to compare the flora of naturally delivered babies versus C-section babies and to see how they're different. I mean, because as soon as you start moving down the birth canal, you start acquiring bacterial flora. And it probably is a little bit different than if you're delivered by a C-section. But I don't know what implications health-wise that could have. That'd be an interesting study. The last one is kind of interesting, and that kind of gets back to that article I told you about that's on the website. Germ-free animals tend to have less body fat. Okay, what's the significance of that? We think that your normal flora help to break down undigestible carbohydrates, for example, and that increases fat storage in the body. So the normal flora is not insignificant, and it really is very, very important. So, let's concentrate on infection. Oh, this slide is just a little bit more detail about some of the common organisms that you can find in various portions of your body: the skin, the GI tract, large intestine, upper respiratory, etc. Okay? I would encourage you to look at that for informational purposes. But again, I'm not going to pull information off that slide for an example. There are several regions of the body, however, that are and should be sterile, microbial free. All of the internal tissues and organs of the body: heart, liver, lungs, etc. And fluids within the body, particularly blood and cerebrospinal fluid. What are we doing when we do a spinal tap? What are we looking for? We're looking for bacteria in the spinal fluid. It should be free of bacteria. And you can actually see it when you do the tap. It should be clear, like a tube of broth should not be turbid. If that spinal tap fluid is cloudy, that typically indicates microbial contamination.
They look for the bacteria, they look for a protein increase.
You're looking for a lot of things, but that's one of the things you're looking at. Okay? You can also tell antibody levels when you do a spinal tap, and that is very diagnostic of various conditions. What do we say we have when the blood has microorganisms from it, bacteria? What's that called? Yeah, septic or septicemia. Okay, and that's a very serious condition. It's very difficult to treat septic patients. I know Lily has been working on sepsis drugs for quite some time. Okay, Some terminology, and I'm not going to read through all of this. You can read this. Some of this I've already referred to. Some of it we will talk about in later contexts. But there's a lot of terminology associated with infectious disease. Okay? So remember what infection means. Infection refers to growth and multiplication of microorganisms in your tissue. Okay? So these would be bacterial and viral induced diseases or fungal. Okay? So the difference between acute, chronic, and subacute, for example, is important. We talked a little bit about asymptomatic. We'll talk later about communicable and contagious diseases versus non-communicable diseases. Types of infections, systemic, focal. Systemic, you know what that one is without reading it? Systemic infections permeate your whole body. Okay? Measles is a great example of a systemic infection. You break out of measles, you just have that measles rash everywhere on your body. Okay? Some tend to be more localized in particular regions. You'll see some nasty pictures of some staph infections that cause boils and things like that. And pimples are a staph infection. Okay. And then differences between primary and secondary infection. So you might want to go through this particular slide and make sure you're familiar and comfortable with some of this basic terminology. One of the things that really is important to know about a particular pathogen is how much of that particular pathogen does it take to typically cause disease. And this gets into a concept that's important called virulence. Virulence is really a measure of pathogenicity. How nasty is the bug? Okay. One of the things that tends to characterize some of the opportunistic infectious agents is that they are not particularly virulent. And as long as your immune system is intact, as long as your normal flora is working properly, they're held in check and they don't cause disease. On the other hand, some things are highly virulent. They're very strong pathogens. And even if you have a healthy, functioning immune system, they can overwhelm that immune system. They are very virulent. Ebola is highly virulent. Okay? You can have a perfectly functioning immune system, and if you get exposed, it can overwhelm you. One of the things that's kind of related to virulence is this concept of infectious dose. On the average, how many organisms of the pathogen do you need to cause disease symptoms? So what's the relationship between infectious dose and virulence? The more virulent you are, the lower the infectious dose. Okay? So if we look at this particular chart up here, the most virulent of all of these disease agents is measles. A single virus can cause disease. Look at cholera. It takes about 1 million cholera cells to cause disease. You've got to slug down a whole bunch of cholera bacteria. Now, if it's living in water, and the water is very contaminated, you can get that 1 million cells in probably a mouthful of water. One of the things we're going to talk about is why are some organisms highly virulent and why are some organisms lowly virulent or even avirulent? If you're avirulent, then if you're growing, you cause an infection, but that infection is asymptomatic, right? So one of the things we're really going to concentrate on are things called virulence factors. What adaptations do pathogens have that allow them to cause disease? we're always in contact with microorganisms. They're everywhere. We've seen it. Okay? But if that contact is going to progress into a disease state, the microorganisms have to gain access to your internal tissues. And typically, that involves some portal of entry. This particular slide illustrates some common portals of entry for pathogens. Some of them are very obvious. Some of them maybe not quite so much. Microorganisms oftentimes are very specifically adapted to a particular portal of entry. And if they are not given access to that portal, they can infect you. The influenza virus is a great example. Influenza is adapted to a portal of entry, the mucous membrane. And so if you inhale influenza virus and they get into your respiratory tract, they can then infect those epithelial cells lining your respiratory tract and you can get sick. Or if you touch the eye and the conjunctiva of the eye gets exposed, flu virus can colonize those cells as well. But I could take a fingerful of gooey substance containing all kinds of influenza virus and rub it on my intact skin, and I wouldn't get sick because they are not adapted to that particular portal of entry. One of the best things you can do during cold and flu season for viral infections, preventing them, is wash your hands a lot. Okay? Physically removing those virus from your surface of your skin. So you then don't touch your eye, or touch your nose, or touch your mouth, and give those virus in a way to enter and get in contact with your mucous membranes. Pardon me? Some microorganisms are adapted to more than one portal of entry, and that can make them a little bit difficult to treat. Tuberculosis has multiple portals of entry. It can be inhaled, but it can also be ingested. Sometimes, depending upon what the portal of entry is, the same organism will cause different diseases depending upon how it enters the body. So you get really different symptoms and different outcomes if you ingest TB as opposed to inhale it. It really does cause a different set of symptoms. One of the things that really is absolutely essential that has to happen if we're going to have infection is that those microorganisms have to cross that portal of entry, and then they have to be able to attach to host cells once they've crossed that portal. No attachment, no invasion, therefore no multiplication, therefore by definition no infection, and therefore no disease. So you've got to have adhesion. We've talked about some microbial bacterial structures that facilitate adhesion. And they include fimbriae. Remember those little Velcro-like protein structures? Fimbriae and some other organs or structures in the bacteria as well, like flagella, have some molecules on their surface called adhesins. These adhesin molecules are microbial, okay? So they might be part of the pilot, for example. But they interact within cell surface receptors on the host, and they bind. These adhesin molecules are called ligands. Ligands are things that bind to other molecules. Okay. Virus have adhesins. Those little spikes that stick out of the surface of many virus, like the HIV virus, are adhesin molecules. And they have to match up with particular cell receptors in order for the contact to be solidified and for that adhesion to take place. Sometimes the pathogen will have more than one type of adhesin molecule. Maybe you've got one on a pilot. Maybe you've got another on a flagellum. Okay? So that means you could potentially infect multiple types of cells, or multiple molecules on you could attach to the same cell. And what's really insidious about virus is these adhesin molecules can change as a result of mutation. That's what we're so worried about in cases like bird flu, the H1N1. Okay? Normally, that particular bird flu virus only has adhesins that match with bird cells. But if a mutation occurs, then those adhesins might gain the ability to attach to human cells. They've jumped that species barrier, and they can now infect a whole new range of hosts. And unfortunately, virus, particularly RNA virus, tend to have very high mutation rates. So the change in these adhesin molecules, that change can happen very, very rapidly. So here's a microorganism with some ligands on its surface, and it's binding to a receptor molecule on a host cell surface. And once you have those cells bound, those pathogens bound to the cell surface, then that is the first requisite step in infection occurring. So, if a microorganism is going to be able to cause a disease state, it's got to become established in the tissues. So in order to understand that process, we need to do a little review. You've all had at least animal physiology. Many of you have had A&P as well. So tell me what a tissue is. What is the definition of a tissue? Okay?
A group of cells working together for a specific job.
Okay, it's a group of cells that are similar in either function or structure. And there's one more criteria to be part of a tissue. The cells must be connected, yeah. Okay, so it's a group of connected cells that have a similar structure or function. Human body has 4 major tissue types. What are they? Epithelial, connective, muscle, nerve. Okay? 4 major tissue types. We're going to be interested in talking about infection about 2 of those 4 types of tissues. We'll talk about epithelial and connective. Those particular types of tissues constitute a big part of our initial line of defense against microorganismal infection. And when we talk about the immune system, I'll make constant reference to your immune system as a series of lines of defense. And we'll talk about the first line of defense, the second line of defense, the third line of defense. And so these are going to be very important in that first initial line of defense. It's kind of like the fort out in the Old West, okay? And if attackers are going to wipe out the settlers, they've got to cross those fort walls first. An initial physical barrier in many cases. So, where do we find epithelial tissue?
Isn't like my organ.
Epithelial tissue is found on all of the body's external surfaces, which include skin, okay? But what else does it include? What else is external to your body technically? All the linings of your tracts and ducts in your body, okay? Your oral cavity, your respiratory tract, your GI tract, your urinary tract, okay? All of those are lined with epithelial tissue. Typically, we subdivide epithelial into two types of tissue, keratinized epithelium and mucosal epithelium. Where do we find keratinized epithelium? Yeah. In terms of tissue, skin, what is keratin? It's a protein, actually, okay? And when that protein becomes deposited in the outer layers of your skin, your epidermis, it makes those cells very tough and insoluble, okay? And makes them a really good barrier. Now, your body's constantly shedding skin. That helps to prevent pathogens from gaining entrance because they attach and then the skin gets rubbed off. So it's constantly renewing new skin cells, and so that process of deposition of keratin is an ongoing process, particularly as skin cells age and they're pushed further up to the surface before they're shed. Okay? Keratinized epithelium is a wonderful, wonderful barrier against infection. But if it becomes compromised, then that's an issue. So cuts, and particularly burns, can be really devastating. Burn victims are incredibly prone to infections. And that's one of the things that really kills most people with severe burns, are just an overwhelming of the body by infectious agents. Epithelial mucosa lines your GI tract, your respiratory tract, and things. Sometimes, in some locations, that epithelial mucosa is ciliated. Where do we find a lot of ciliated epithelial mucosa in your body? Respiratory tract, particularly your trachea. And what is the purpose of those cilia? Yeah, they beat back and forth and they tend to sweep particles back up out of your respiratory tract. It's referred to sometimes as the ciliary escalator. Okay, and it creates an upward sweeping motion. People who smoke have a higher incidence of lung infection. How come? Go ahead, Audrey. They damage these cells, and the nicotines and things in the cigarette smoke paralyze the cilia. So you're compromising that particular defense mechanism. Epithelial tissues also contain goblet cells. You know what goblet cells produce? Mucus. Okay, those are the mucus secreting cells. What's the advantage of producing mucus?
Actually, it's crap.
Yeah.
It pushes stuff out so stuff can't go.
Essentially, yeah. I mean, it helps to work with this ciliary escalator. What the mucus does is it basically kind of slimes the pathogens and covers up some of those adhesive molecules and prevents attachment, so they're easier to expel by either just this or a combination of coughing and sneezing as well. So what does your body oftentimes do in response to infection? You speed up mucus production. Okay, and that's an attempt to